All right, these are your RNA notes, and it'll actually talk about RNA, the genetic code, which is right here as our title, and then transcription and translation. So the genetic code. So DNA and RNA in general are read 3 bases at a time. This is called a codon. So a codon is a 3-base sequence right here that codes for specific amino acids. So it could be something like ACC, or let's see, if it was DNA, it could be TAG. Anything that's just 3 of the letters. There's 64 possible combinations. So as a quick review from the biochemistry unit, amino acids together make proteins. And there are 20 different amino acids. Most of you, as one of your projects on your choice board, have to do a project on one of those amino acids. So a breakdown of codons. So if you take a sequence, so right here we have a sequence, it just looks like a bunch of letters. Then you just separate it into codons. So every 3 letters, you put a space, and it ends up having these dashes in the middle, and that's how it's typically written, or you can just write it with spaces. And then later, we'll use these to identify what the correct amino acids are. But first, let's talk about the structure of RNA. So RNA is a ribonucleic acid. So we lost the D, so now it's just ribonucleic instead of deoxyribonucleic. And it has the sugar ribose instead of deoxyribose. Bases are, we still have A, we still have G, we still have C. The big difference here is your uracil, or U. There's no more T, no more. So they pair as follows, and this is where it changes again. A and U pair together, and then G and C still pair together as before. Another big thing about RNA is it's single-stranded, so it's just one single strand. And even more special, it can leave the nucleus. DNA has to stay in the nucleus, so RNA is special because it can leave it. So there's 3 types of RNA, so there's 3 total. You have messenger RNA or mRNA, and to be quite honest, we'll really only refer to it as the little letter abbreviation, not messenger. Then you have RNA, or rRNA. Then you have tRNA. So mRNA is what carries the genetic code. This one leaves the nucleus. rRNA joins mRNA and tRNA. And tRNA is what transports the amino acids. And I'll show you some diagrams, so don't worry. So here's your types. Right here, this is showing mRNA forms from the DNA template strand. tRNA is right here with the amino acid attached. And it comes into the ribosome, which is where the rRNA is also located. So where does all this happen? Like I said, mRNA moves from the nucleus to the ribosome. rRNA is in the ribosome, and tRNA is ribosome to cytoplasm. So if you look at it as kind of like a little train, the way it goes makes it stops. So it starts in the nucleus, it goes to the ribosome, and then it goes out into the cytoplasm. So now transcription. So transcription is making RNA from DNA. So you have an RNA polymerase, and this term shouldn't be new to you, except in DNA it was DNA polymerase, and it binds to the DNA. So right here, you see our RNA polymerase. It binds to the DNA and makes a complementary strand of mRNA. You can't just say RNA, you have to say mRNA. And there's a little typo here. That should be a U, not a T. So C and G still pair, but now A and U pair. So it's the same thing that we did where we had the strands written and it said like A, T, G, C, and you below it you wrote the complement. You're going to do the exact same thing, except now wherever you would write a T, you're going to write a U. And why is mRNA used instead of a strand of DNA? Simply because mRNA can leave the nucleus and DNA can't. So introns and exons, are something that we're going to talk just briefly about. But sometimes the code is interrupted by these sequences that will not be in the final mRNA. So the ones in between are called introns, and the RNA that you actually want is called exons. So the introns are cut out by enzymes, and the exons are kept. So if we look at this picture, all the yellow spots are exons, but this whole thing is a strand. Well then, during transcription, we get rid of all these blue spots here, to where eventually we get just this strand of mRNA. So transcription practice. So this is pretty simple. You'll have to do this again in class. But so A normally would pair with T, but because it's mRNA, we're going to write a U. G still pairs with C, C pairs with G. Now T will still pair with A because this is mRNA. DNA is allowed to have a T. C is G, C is G. G is C, A is U, T is A. G is C, C is G, A is U. T is A is U, C is G. TT goes with AA, C, and then G, G, U. Okay, the next one. Once again, mRNA. So no T's should ever be in this strand, ever. Only use A's, G's, and C's. So G and C, C and G, G, and we'll have U, C, A. We'll have C, G, A. Then we'll have A, U, G, C, U, U, G, A, C, and U, C, A. All right, so now translation. So long story short, this is transcription. It's making mRNA from DNA. It's a pretty simple process. Translation is a little bit different. And the way to remember the order is if you remember replication, and then we have transcription, and then translation. Okay, alphabetically wise, R comes before T. So this is first. But then if we look, TRANS is the same, right? But if you look at, let me change my pen. If you look at the C here and the L here, what's first alphabetically? Well, the C is. So, because C comes first alphabetically, I always remember that transcription is second, translation is third. And this is something you will have to answer. You'll be asked the correct order of the processes. So translation. Here's kind of a breakdown of what happens. So after transcription, mRNA leaves the nucleus. Then it attaches to a ribosome. Now tRNA brings the proper amino acid. So tRNA is kind of this funky little like T-shaped thing, and it has an amino acid attached to the top of it, and it has 3 bases on the bottom. So tRNA will bring the proper amino acid to the ribosome that codes for these 3 letters. And amino acids will form a peptide bond, eventually forming a polypeptide chain of amino acids, also known as a protein. Remember, poly means many, so it means many peptides. Then tRNA also carries the amino acids in the form of an anticodon. The anticodon is this section up here. It's those 3 bases that match with the RNA. So just think codon, anticodon. So now there's 3 sites in the ribosome when we're doing this process. So you have the A site, the P site, and the E site. So A is amino acid site, so that's where it goes in first. A is the first letter of the alphabet. A is where it goes in first. Simple. P is where the tRNA enters the protein. Okay, so it binds during A, and it enters the protein during P, and then E stands for exit. So it goes in for A, first letter of the alphabet, it goes out for exit. And what happens is, that funky little thing I drew, the amino acid goes away, and you just get the structure with nothing attached to it. So here's kind of elongation how it happens. So if you look, this is the first amino acid that came on. This is the second. The third is in the P site. This one's in the A site. So now, as it moves down the chain, it attaches. So if you notice, the 3 and the 4 aren't attached. But then if we go this way, it's moved down the site. So this is where I said it's kind of like this little naked tRNA. It doesn't have the amino acid. So that's the exit site. And now this one is bonded. They're bonded together. And then it goes this way, and that one exits. So that one's going to exit, and eventually another one will come in here, and this one will move down. So it's this continual process. And here's just another picture. This is actually the same picture that's in your book. So it shows you that transcription is up here. So you see it right there. And translation is down here. All right, so now the fun part. So this is a basic codon table. And they're kind of tricky to read at first if you've never done it, but once you get the hang of it, it's really simple. So there's only one start codon. So your chain cannot start unless it has AUG. It absolutely has to have AUG to start. Otherwise, it just keeps going until it finds AUG. So AUG is on the chart right here. And how we read this is you go to the first base, the second, and the third. The first goes this way, the second is across, and the third is down. So there's stop codons as well. So when it finds a stop codon, it ends. So the stop codons are all right here. Okay? So here's your stop codons, start codons. So really simple how you would read it is you go down and across. So let's, I don't know, we'll do a random one. Let's do CAG. Okay? So first you find the first base first, so C. So we're only going to look in this category now. Okay? And because it's, the second letter is A, so now we're only looking in this category because it goes down from the A right there. And then G is our last one. So G is right here. So CAG is glutamine. And that's kind of how you read it. And don't worry, we will do tons of practice with this. So, translation practice. Now here's where you take what you see in the chain and then you use it to translate into an amino acid. So with this, how it's going to happen is we're going to take the first one and go from there. Now you know, or hopefully you will know soon, that AUG is the start codon. So you have to say, you can say methionine is what it is, so it's MET, or you can say start. It's up to you. I'm just going to say start. Then you would need to go back to the table and use the 3 letters to determine the next one. So CCG is proline, or PRO, and we'll talk about abbreviations too. UCU is serine, or SER. AAG is lysine, or LYS. And then UGA is stop. So now we've just made our amino acid chain. So let's do another one. So AUG is start. GCG is alanine, ALA. AAA is lysine. And if you notice, these can code for different things because AAG is a lysine, but AAA is also a lysine. UAA is a stop. So you know what? We don't have to do that one because we stopped. The next one, well, CCC, that's not a start codon, so don't do that one. AUG, there we go. There's our start. Now we have GAG, which is glutamic acid. I don't remember the abbreviation, so I'm going to do it like this. And then UAG is a stop. So now we don't do that one either. So if you notice, not every time you do this will these chains be exactly the same length. And here's just another diagram to kind of show you all these processes. This line right here is symbolizing the nucleus. The cytoplasm's out here. All right, so now mutations. this is when things go wrong. And sometimes there are things that go wrong that happen and cause problems. So what is a mutation? Really important to note that it's a permanent change. And it can occur spontaneously or from chemicals or radiation. So there's lots of times in history where there's been a radiation issue where people all of a sudden have all these mutations, or there's frogs born with 3 legs, just weird things like that. But there's 2 main types. So you have gene mutations and chromosomal mutations. So gene changes a single gene. So your DNA strand has just one thing is a gene, whereas the chromosome would be that entire chromosome and all the genes in that chromosome. So, quick review of cell types. Hopefully this is very simple for you to remember. We just did this. Somatic cells are body cells, and sex cells are reproductive cells. So mutations in somatic cells are blank on to the next generation. Well, we kind of talked about this. You did have an essay about this. Somatic cells, they are not passed down. This is your body cell. This is like you have a scar. Your kids aren't going to have that same scar because it's something that happened to your body cells, and those genes are not passed down to your offspring. Whereas mutations in sex cells are passed on to the next generation. And why is the above true? Like we said, your sex cells only have those 23 chromosomes. They have to pair up. They have certain genes that give you the traits that you have. So different types of gene mutations. Now we're just talking about the ones that are just an individual gene. You have a point mutation. Simple way to remember this, a point mutation occurs at a single point. So it's one of the letters. So maybe a substitution. So think about it in a sports game. If there's a substitution, there's one player going in for another. So a single nucleotide is substituted for another. So in your chain, you might have a G instead of an A. And if that was a start codon, that could be a problem because it won't replicate. If that was somewhere else, it'll be a different amino acid, and it won't make the same protein that your body needs. There's also a frame shift. So think about shifting a frame, like something's taken out, so everybody has to shift down a spot. So if you're in a line and somebody leaves the line, you all have to shift down. Same thing, if you're in a line and someone cuts in front of you, everybody has to shift the other way. So think about it this way. So an insertion, plain and simple, when a nucleotide is added, once again, that's all your letters A, G, C, etc. Okay? Deletion is when one is removed. So that's when the letters are just plain, flat out removed. So here's just kind of showing you what can happen. So a gene mutation, here's your normal, here's one with mutations. So you have the letters changed. Now look what happens in a frame shift. You have start, or met as your start, and you have glutamine, alanine, glycine. But now that G is gone. So now everything shifts. So now it's AAG, CAC, and this one doesn't have its third letter. So now we have start, but now we have lysine and histidine. Those are going to be amino acids that don't code for the same protein that your body needs. All right, and then this chart, just pause your video and kind of look it over. We'll go over this in class too. We'll do a lot of practice with mutations. This kind of shows you using this analogy sentence here. The big fat cat ate the wet rat. And it shows you what happens when you change letters. So a nonsense substitution, the big rat. A deletion, that one's really messed up. This one, insertion, changed everything. This one, a duplication, the big fat cat. See how this changes? So look through all these things, and then really cool over here, it tells you examples of diseases that happen as a result of this happening. And now chromosomal mutations. So these are changes in the number or structure of the chromosomes. So a chromosome, here's your centromere, okay? So there's 4 types: deletion, duplication, inversion, translocation. So deletion, you should know what a deletion is. We just get rid of something. So in this example here, the B is gone. So now it's different. A duplication, when you duplicate something, you're making a copy. So in this one, we have another B, so we added a B. So now you have 2, and now it's different. An inversion, if you invert something, you're turning it upside down or changing it. So an inversion is just changing them around. So it used to go A, B, C, D, E, F. Well, the A and the F are still in the same place, but look what happened. C and B switched places with E and D. And then a translocation, that's when two chromosomes actually switch arms. So this is kind of like crossing over, although this is crossing over when it's bad. So this is when crossing over results in bad things. So if you look, A, B, C, D, E, F, G, H, I, J, K, L. Now we have A, B, C, J, K, L, and G, H, I, D, E, F. And a little bit of mutations practice. So it just kind of shows you what happens. So, and we'll kind of just look at the mRNA. So this used to be CGTAC. So the mRNA would be GCAUG. Now it's going to read GAAUG. So see what happens when we substitute it? It's totally different. Insertion, one or more base pairs are added. So we added an extra A here. So it used to read GCUACC. Now it reads GCUACC. And then deletion, when one or more are lost. So we lost our G right here. So it would have been GCUACC. And now it's GUACC. And that's the end of your notes.